Hello, welcome students. We shall start a new chapter, that is molecular basis of inheritance. We shall start a new chapter, that is molecular basis of inheritance. Molecular basis of inheritance. You know that inheritance means transmission of characters from parents to offspring. But who is the molecular basis of that? Actually, you know that DNA, that is deoxyribonucleic acid, that is deoxyribonucleic acid. About DNA you have already studied something in class 11 and your other classes, and we know that DNA is the genetic material, means carries characters from parents to offsprings, and although the second nucleic acid is ribonucleic acid, it is also the genetic material in some meiosis. So we have no need to take more attention on this. We have to take our attention on that DNA. DNA is the genetic material. First you should know what are the necessities for the genetic materials. How we can say anything that it is a genetic materials. It has following features. It has following features, that it should be capacity to replication, means it has capacity to replications, means capacity to produce its replica, means it has capacity to produce its similar, or not identical but similar types of parts. So capacity to produce its replica has mutation property. You know about that mutation is sudden and permanent change in the gene. So it has ability to undergo mutation. The mutation of may be harmful, may be useful. अब ये जो म्यूटेशन होता है वह हार्मफुल भी हो सकता है और यूजफुल होता है. This useful mutation help in evolution. So that is help in evolution. So mutation, so property of mutation should be found in that. Second, chemically and structurally should be stable. Its chemical nature and structurally should be stable, not unstable. So it is also another feature of the genetic materials that it is chemically and structurally should be. And third, it should follow the Mendelian concept, means it can introduce in itself to express the Mendelian property, means ये अपने आप को Mendelian property दिखाने की स्थिति में भी अपने आप को ला सकता है. So it should follow the Mendelian concept. तो ये these are the some properties, some features. If that is found in any, that will be called genetic material, and we will see that all these features will found in the DNA. So DNA is also known as the genetic material. Now the nucleic acid. Nucleic acid, it was first discovered by Friedrich Mecular in 1869 from pus cell. From the pus cell it is discovered. From the pus cell it is found. Friedrich Mecular discovered, and but he gave the name nuclein. He called it nuclein. Later Altman called it nucleic acid. Altman called it nucleic acid, and it is of two types. It is of two types. One is DNA. Next one is RNA. DNA, that is deoxyribonucleic acid, and this is ribonucleic acid. This DNA considered as genetic material, and this RNA mainly related to the protein synthesis. Understood? So two type of nucleic acid. Each nucleic acid, whether it is, means this is the content. Each nucleic acid made up of what? That is nitrogenous base plus sugar plus phosphate. Each is composed of, nucleic acid is composed of nitrogenous base, sugars and phosphate. That is called phosphoric acid. These two are called nucleoside. These two, nitrogenous base and sugars, they are called nucleoside. And nucleoside plus phosphate is combinedly called nucleotide. So we can say that nucleic acid is the polymer of nucleotides. Means there are so many nucleotides arranged to each, and thus that nucleotide or the nucleic acid is formed. Now, thus one thing you know, as you see that these are the more or less same. Now, I have this sugar. In the DNA, the sugar will be deoxyribose. In the DNA, the sugar will be deoxyribose, and in case of ribonucleic acid, that is the sugar will be ribose. So two types of sugars we get. One is deoxyribose and next one is ribose. This is found in DNA. This is found in RNA. So we will consider about that. One things, deoxyribose and this is ribose. These are five carbon compounds. Five carbons are compound in that, and their structure will be like that. This is the first carbon, this is the second, this is the third, this is the fourth, and this is fifth carbon. Understand? In the same way, this is the first carbon, this is the second, this is the third, this is the fourth, and this is the fifth carbon. So fifth carbon compounds are found. In that, all the carbons there is will be H, OH, with first position carbon, this is C, HOH, 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 HH, and the fifth CH2OH. In the same way, this is the main difference between that the second position this is OH. Understand? The second position contain of ribose, two functional group, that is OH, 2 moles of functional group, that is OH, and in that only one OH. So it is more reactable. Means because it contains two functional group and it contains only one, so it is more reactable than this. So it is DNA in comparison to the ribonucleic acid is less reactable, that is like a stable. And the structural formula you see. So this is the you know that first carbon, second, third, fourth, fifth. So this is the 5 carbon compounds. This is the ribose and this is the deoxyribose. Understand? And now this ribose and deoxyribose sugar is attached with one side with nitrogenous base. It is attached with nitrogenous base. Nitrogen base means which contains mainly nitrogens. This nitrogenous base is of two types. One is purine, next one is pyrimidine. In this, the two components are found. One is adenine, guanine. In this three compound, that is thymine, cytosine and uracil. So there are 2 and 3, they are found in that. Now this is this sugars, this is the nitrogenous base, and now the phosphate phosphoric acid, that is H3PO4. Now the H3PO4, that is this is the phosphoric acid. So these three nitrogenous base, that is sugars, and so these two combines to form nucleoside, and all the three means with phosphoric acid plus this one, they all three will combine and forms a monomer, and such type of monomer is called nucleotide. And so many nucleotides combined to form nucleic acid, and these are the adenine, thymine, guanine. These are the called bases of the nucleic acid. So, and one thing you know, all the whether it will be DNA or that will be RNA, they contain 4 base. Adenine will show by A, guanine will shown by G, thymine will T, cytosine will C, uracil will U, and the base pair will be A, T, G, C, A, U, G, C. So this means only 4, 5 will not use the 4. And place of thymine there is uracil. That is the main difference between the base of deoxyribonucleic acid and ribonucleic acid. And how they arrange we shall discuss in the next video.